Hi, everyone. I am Stacey Pike Langenfeld, president and co-founder of Krabbe Connect. Thanks so much for being with us today. In honor of leukodystrophy and Newborn Screening Awareness Month, I am wearing my earrings to showcase my support. We have had a series of webinars this month, really around knowledge about the treatments, the different types of transplants, the caregiver experience navigating Krabbe disease, and now an update from our one and only current clinical trial in Krabbe disease called ReKlaim, sponsored by Forge Biologics, which is now owned by Ajinomoto. So I'd like just to take a moment to introduce Dr. Maria Escolar. She is the Chief Scientific Officer for Chief Medical Officer for Forge Biologics. She comes to the space with significant experience in Krabbe disease and spent a majority of her career helping those families and diagnosing them, also starting the NDRD clinic at the University of Pittsburgh Medical Center. With that, I am going to go ahead and turn it over to Dr. Escolar to further introduce herself, as well as start her presentation on the update on ReKlaim.
Thank you so much, Stacey and Krabbe Connect, for inviting me and giving me this opportunity to talk to you about Krabbe disease, what we have learned over the years about the standard of care, which is umbilical cord blood transplantation or bone marrow transplantation, and then FBX-101, the trial called ReKlaim, which is a new type of gene therapy. And I'll start by saying that, disclosure statements are that I was the inventor on the patent application describing FBX-101 that was licensed to Forge Biologics by the University of Pittsburgh. Forge is a subsidiary of Ajinomoto, as Stacey explained. And I am sure you're all very familiar with what the inheritance pattern of Krabbe disease is, but for those who are new, it's what we call autosomal recessive, which means that each parent carries a mutation, and you have 1 in 4 chances of inheriting both mutations, and this is when you develop the disease. This disease can present very early, even at birth, but it can also present when you're an adult. These mutations sometimes are known and described very clearly, so you know what they mean, but many other times they are not, and they're called variants of unknown significance, which means that there is an abnormality in that gene. However, people don't understand if that causes disease or not. And it's quite complex because there are other small mutations that are common in the normal population that we call polymorphisms that can further decrease the level and then cause disease depending how they're combined with that mutation. So one of the most important things is to actually create databases of mutations, because that's really important to try to predict what is going to be the onset of disease when you pick these up through newborn screening, which I'll talk a little bit about later. But first of all, I wanted to tell you that there is a mouse model of Krabbe that recapitulates the disease very closely to that of the human, and it's called the twitcher mouse. And so here in this figure, I'm just trying to portray what happens. What we call the wild type is the normal mouse. So the normal mouse, you can see how the neuron looks, or the oligodendrocyte looks pretty normal as it starts developing, and then it develops all these branches or ramifications, and then you develop myelin around it. But in the twitcher mouse, which has galactocerebrosidase, this enzyme that is deficient, you can see how the myelin gets disturbed. And over this time of maturation, or how they, let's say, differentiate into mature cells that make the myelin in the brain and peripheral nerves, when you don't have galactocerebrosidase, you accumulate another chemical that is called cytosine. You just can't break it down. And so over time that increases, and then you go into a mechanism that we call apoptosis. What that means is that cells die. And unfortunately, if this happens towards the end of pregnancy, like after 26 weeks of pregnancy or even after birth, it has a huge effect. And I'm just showing you on the right how myelin looks like. So you have different type of cells or neurons, I should say, in the brain. We call them oligodendrocyte. In the peripheral nerves, we call them Schwann cells. The Schwann cells make the myelin in the peripheral nerves. And that's what I'm showing you here, how it wraps around the nerve. And this myelination is what makes a conduction of an impulse go really, really fast. So when this is destroyed or when these cells die, this conduction suffers a lot, and that's why we start seeing the symptoms. Unfortunately, babies who start with this disease, in average, die by 2 years of age, although there are some that can live longer. It depends how much you take care of the patient and what type of mutation they have. What you see here abbreviated as hematopoietic stem cell transplantation, it includes bone marrow transplant, it includes umbilical cord blood transplant, and I explain a little bit about the difference with those, but we have learned that this can extend lifespan. However, as it extends the lifespan because it really halts the progression in the brain, but the demyelination in the peripheral nerve, which is again the nerves that I'm showing here, which is the ones that are going to your muscles in both upper and lower extremities. As that continues to progress, the muscles become very, very weak, and you eventually die, usually by teen years, some of them late teen years or very early adult years. So we're able to extend the life by a lot. It's a quite effective treatment, but unfortunately, most of the children that now we have followed for quite a long time, almost 26 years, we have seen that there is progression of this peripheral nerve disease. So for those of you who are not familiar with umbilical cord blood, and I'm going to talk a little bit more about this because one of the biggest challenges we have with treating Krabbe disease is that the disease progresses very, very fast, and I'll explain you why. We need a rapid source of transplant. And so when you have a bone marrow donor, it usually takes months to get this patient that matches immunologically with your baby to come in and donate, the bone marrow and then give it to the baby. So because this takes months, we don't have that time. So usually we use as a source umbilical cord blood, which is blood that is drained from the placenta, and then it's taken to a lab and it's prepared and it looks like this bag. It's like 30 mLs. And these pictures are courtesy of Dr. Joan Kurtzberg. But this is the source that we most of the time use, and it has many advantages, especially once you use gene therapy, because it's very immunologically, which means your how you make antibodies against, let's say, things that you haven't seen. This type of immune system is very naive because it's of a baby, hasn't seen anything yet, right? So that gives us some advantages when you inject them with a gene therapy, because they mostly don't recognize the protein that they're missing. They could reject it because they don't see it because they have the mutation, right? But if you change the immune system using umbilical cord that is healthy, meaning that this baby actually had GALC, then when we inject a vector, this gene therapy that we're injecting, your body doesn't reject it. Like your cells from the umbilical cord, they don't recognize it because they're very naive. So that's one of the advantages of using umbilical cord. It has many others, but that's, I think, the most important. Okay, so back in 2005, we published a very important paper which was showing just the short-term outcome of babies that were transplanted. And they were transplanted mainly at that time because they were siblings. So mothers have a baby that was affected, and then they had a sibling that was pre-symptomatic. Neurologically wise, it was, I believe that it was going to be too late for those babies who were symptomatic, given how they were affected neurologically. But at that time, we didn't really know if the transplant could help or not. So some patients were transplanted with symptoms, and those are the ones that you see in the black lines. So each patient is a line there with a color, except the black ones, which were symptomatic. And the ones in color are each patient that was a sibling. So you can see the difference if you have symptoms or no symptoms. And just to orient you in this chart, you're having the calendar age. So let's say if you're 1 year old plotted against, let's say, the developmental age of a 1 year old. So you should be in that green middle line if you're average, and in the gray line if you are within the range of the population, right? So you're seeing that some of those kids are completely normal, some of them are slightly below normal, and then the ones that are very abnormal are the ones that are symptomatic. So what did we learn about this study? We learned that when babies had symptoms, it was too late to benefit from the therapy because we didn't see absolutely any changes that were positive. However, if the baby was a sibling, so it was diagnosed right after birth, we were seeing improvements, not the same in everyone, but most of them were developing normal cognitively, and their understanding of language was also normal. And we were seeing huge improvements in what is called adaptive behavior, which is the ability of the child to take care of themselves, as well as in expressive language. So that was kind of the first observation we had. So we decided, okay, now we have to focus just on the pre-symptomatic ones because those are the ones that are deriving a benefit. Obviously, we wanted to treat the ones that came late and were symptomatic, and that's one of the reasons why I started that program called NDRD, to be able to provide also services to those patients who were symptomatic, because they also can be helped, but not in the way that we could help the ones that were asymptomatic. We continue to study this because some of those babies that, even though they were pre-symptomatic, they were still having a little bit of disability, and we didn't understand why. And we realized that even in those cases, we could actually see some abnormalities in the brain MRI, in the nerve conductions, or in what we call the auditory brainstem responses, which is kind of how you conduct, but through the auditory nerves. And we realized that some of them had progressive spasticity, which is tightness in the muscles. And the blindness, deafness, and inability to eat by mouth, that was a much later symptom. So with the transplant, if you do it really quickly before they have more injury to the brain, you could actually prevent some of the deafness and blindness. And then as these kids grew older, we started seeing some muscle atrophy. So their muscles became weaker, not as strong. And then we saw that they developed this, what we call peripheral nerve disease. So again, those Schwann cells started dying in the nerves that go to the muscles. Okay, so we then found out there are two different problems. One is the brain. Why are we able to stop the demyelination in the brain? Well, that's quite amazing, actually, because if you look at these pictures that I show you here, what you see in blue initially is myelin being developed from when you're about 107 days after birth, so approximately 3 months, which is, as you know, when most of the time the disease starts. But you can see in color as these babies grow up all the way to 179 days. 329 days, which is almost a year. So up to approximately a year of age, you can see how much myelination you produce. So it's more than ever in your entire life. So, okay, the transplant works because the healthy cells are able to donate the GALC to the cells that don't have GALC. And we were able to stop the demyelination. But what could we stop? Not what happened already, but what would happen. The myelin that will come in the future will form normally versus abnormally. So it was still a huge effect in the brain. But somehow, as these babies grew and got older and bigger and their extremities grew, it wasn't enough for the peripheral nerves. And just so you understand how different the disease can impact you as a newborn, here I'm showing you a tool that we developed to understand this at birth. And we did brain MRIs of a lot of babies. All these dots in blue are normal babies. And the ones in red are babies that were diagnosed with Krabbe because of a sibling. So you can see that some of the red dots are really, really low, and other ones are kind of overlapping with the low area of the blue ones. And so what that told us was that some babies, even at birth, were almost 6 standard deviations in myelin below normal, while other ones were close to normal. And this is at birth. So the disease starts before birth in many babies. In other ones, it's right after birth, but it's something that develops extremely fast. And why does it develop this fast? Because of what I just showed you of the brains myelinating so quickly. So in my view of Krabbe disease, when you diagnose a baby at birth, is a medical emergency. You have to get this baby treated as fast as you can because every single day makes a difference. We learned that what's lost cannot be recovered. You can only prevent loss, but you cannot recover what's lost because those cells die. Okay, so we've been fighting and trying to get newborn screening approved since those days. So again, that publications was in 2005. And as you know, Krabbe newborn screening was submitted to be evaluated by the RASP back in 2009, and it was rejected. And then it was just recently approved. We're very happy about that. There are 12 states that are screening, and many more that will now that was recommended. Many other states are going to join, and it's something that is really critical to be able to treat this disease for what I just explained. So we really have to identify these babies at birth. Now, there are some challenges because when we identify them, sometimes we can say, in the majority of cases, we can say they're going to be infantile, but sometimes they are late infantile, sometimes they're juvenile, sometimes they're adult. However, we have a biomarker, which is the cytosine, what I explained you, these babies accumulate, and we've gotten really good at being able to predict the infantile form. And so you will hear about cytosine also a lot when people talk about Krabbe, because that is a really good biomarker for the very severe form, the one that is really a medical emergency. So what happened after that? So I started thinking, okay, what else can we do to increase the levels of galactocerebrosidase so that there is enough so that the peripheral nerves can also benefit from this? And so we developed this therapy. The name of the clinical trial is ReKlaim. You may have heard about Rescue. We used to have it in two separate trials, but then we merged them into one, and we just call everything ReKlaim. And I'll tell you a little bit about that. But the way we chose to treat was intravenously. Before we decided this, we looked at it in all the different ways: intraventricular, intracisternal, intrathecal, in the mouse model. But we realized, especially when you think about how this can be really translated into humans, so a lot of these experiments were done in mice, we realized that we had to do this intravenously. So we do it after umbilical cord blood transplantation because we also learned through some mouse experiments that you actually need the healthy immune system to really develop normal peripheral nerves. And so that's why it's done one after the other. And one of the experiments that was done by Dr. Wenger, who was the one leading the mouse, the Twitcher studies, in this figure here is showing you the survival of the mice, and the numbers below are kind of the days they survive. So you have the purple one is the untreated mouse that dies at about 50 days. And you can see that if you give bone marrow transplant alone or gene therapy alone, you don't see much of a difference. But when you combine the two, which is the blue line, you see this synergistic effect in which there's huge improvement. And this is because of some of the things I was explaining before, that you need the effect both in the brain and the peripheral nerves, and you need a healthy immune system. We then treated dogs. And so what we saw was that if you look at the untreated dog, and dogs have Krabbe disease also as a natural mutation, this dog was, reached what we call humane endpoint, meaning that they have to be hacked because they're suffering at 14 weeks. When we treat it with just AV only, you can see that this dog is struggling a little bit too, but is able to eventually stand up and walk. But there's a lot of weakness in his extremities, especially the back ones. And this is the dog that was treated with both bone marrow transplant and AAV. And this was using a high dose of AAV. Okay, so that's what led then to the trial that we call ReKlaim. And it was done creating what is called a vector. And a vector has the outside of it. You may be familiar with like coronavirus that has spikes outside. Well, the outside is what we call the capsid. And inside this vector, there is a piece of DNA. These vectors can be, so the capsid can actually be designed so it has preference for tissues. So in this case, we wanted to have preference for the peripheral nerves. So it was designed to do that. It can infect or get into the cells in pretty much any cell in the body, but it prefers to go to the peripheral nerves. And the bottom just shows kind of how this DNA is put together, but what we replace is the GALC gene, the one that produces GALC. And so we want this to express high levels of this enzyme. And so then we had to design it so that we first had the transplant, preferably umbilical cord blood. It gives you the normal immune system because the umbilical cord has stem cells that go into the bone marrow. In the bone marrow, you have red cells and white cells, and these white cells are the ones that let's say we can become immune system, and they are able to recognize anything that your body has not seen. It has the advantage that if you are born without this GALC, you will accept it if you get it through the gene therapy because it doesn't recognize it as let's say as something foreign. And then this GALC is taken by the deficient cells, and as I mentioned before, the umbilical cord is naive to it. And as I told you, you can design it so that it goes to the peripheral nerves. Okay, so then we thought, well, how can we then use this? How can we separate all the different presentations of Krabbe? So we thought, okay, the most urgent need is the newborns because they have the most severe disease. So we tried to focus on them, but we also decided, okay, they have to be asymptomatic. However, we learned from doing some studies in late infantile, which are kids that develop disease after a year of age, that actually the transplant was helping them if they had symptoms, but they had to be very minimal. So these were the two groups we wanted to target. But both groups had to be transplanted. So what you see in this figure in gray is that the two groups, the infantile and the late infantile. So after you have the diagnosis, because you have low enzyme, because you have cytosine testing and you have genetic testing, and you can tell that these are going to develop infantile versus, which is 80 to 90% versus the other ones that are going to have late infantile. There are different, let's say, roads that you could take. If you have infantile and you were diagnosed after symptoms, unfortunately, you cannot get the treatment. If you are an asymptomatic late infantile, you're not going to get the treatment because those kids actually have really good outcomes, even with the peripheral nerves. However, if you are asymptomatic and have the infantile, or if you are minimally symptomatic and you have the late infantile, and you're transplanted, and you're able to qualify, and we'll go over what qualify means, you're going to get then the FBX-101, which is the gene therapy. And then within these gene therapy groups, we have two. And why do we have two? Because we wanted to help as many kids as we could. And the ideal scenario, and that's how the whole trial was designed initially, was to infuse it intravenous during the period where the kid is in the transplant unit and is what we call myeloablated. So they get chemotherapy. You've heard about chemotherapy. What it does is that it kills the cells that are in the bone marrow, which are the ones that as I'm saying, make red cells and white cells. And why did we kill those cells? Because we have to create space for the new cells, the ones that come from the umbilical cord that are injected intravenously. And those cells then go into the bone marrow, and they are stem cells, and they create new cells. And these new cells are from the donor. They have the enzyme. They can donate the enzyme. Okay, so this is what we call the myeloablation group, because again, they had to be myeloablated to get the transplant. And then there's another group, and these are all these kids that already got the transplant, and they went through the myeloablation, and now they're just immune suppressed because they want to make sure they don't reject the transplant. Well, some of them have even stopped the immune suppression. Now they are out in the community and they are with everybody else, right? But now they want to be helped because they have developed peripheral nerve disease. So then we bring them and we immune suppress them. And that's the group we call the immune suppression group. So it's done either after you recover the immune system, or maybe right after you go out of transplant where you are still in immune suppression, And we then decide how much we need to add to that immune suppression and to be able to give you the gene therapy. So these are the two groups. So one of the requirements is that the gene therapy has to be administered at least 20 days after the infusion of the umbilical cord. So after 21 days, you're usually in the unit. After about 2 months, you're outpatient, right? So it could be any time after 21 days. But what happens if it's after 21 days? You have to be at a clinical trial site so that it can be administered. So some babies have to be transplanted in the site where you want to administer the gene therapy because it has to be done during transplantation. However, if your child gets transplanted in a non-clinical trial site, then they can only join the immune suppression group unless would be transferred when they are still inpatient, so from a medical inpatient unit to another medical inpatient unit. So those are all the different possibilities. Okay, so what have we done until now? We have treated 5 subjects. They are all in what we call the low dose. So the low dose means that the number of vector genomes that we give per kilogram. And so we give 1.6 times 10 to the 13 vector genomes per kilogram. All these subjects were diagnosed by newborn screening and received umbilical cord blood transplant. 4 of these were treated with the reduced toxicity regimen. I'm sure you heard about this, so I'm not going to go into any of this except to say that it is less toxic to the organs when you use these reduced toxicity regimens, which means the babies tend to do better because at least that toxicity they don't have to deal with. And so it has many advantages. The disadvantage that it has is that they may need a second transplant, or they may reject that because it's not as toxic, but that's a minimum number of patients. And if they require a second one, they usually then engraft. And because it's reduced toxicity, they usually tolerate the second transplant within months after. And at least we know that we're not damaging the organs or the kids are dying from toxicity. The myeloablation has 2 patients and the immunosuppression has 3. The myeloablation has 2, one 24 days and the other one 29 days post umbilical cord blood transplant. And in the immunosuppression arm, 3 were those, one at 51 months, 17 months, 23 months post umbilical cord blood, and they receive an immune suppression regimen based on these immune suppressants, rituximab, sirolimus, and prednisolone or prednisone. Okay, so let's say you have a baby diagnosed because of newborn screening, or let's say it's a sibling of another one, or there's family history and they get diagnosed at birth, or they are the late infantile that are minimally symptomatic. So what needs to happen? You have to have a referral to a transplant center that is a clinical trial site within 1 or 2 weeks. And those sites are right now the University of Michigan Medical Center or Duke University. That is if you want the child to be treated while they're being transplanted, so they're already myeloablated and you don't need to do a lot of additional immune suppression. Preferably, they would have umbilical cord blood transplants for the reasons I explained, or they may have a bone marrow transplant. Usually, the only way we can get that really quickly is by a sibling donor. But so most of the time, it's going to be umbilical cord blood. They also have to qualify for the inclusion criteria. It's a complicated inclusion criteria. If you want more information, you can go to this clinicaltrial.gov study where it explains all the criteria. But I would only talk about one of the most important ones, which is the antibody testing. So if you've been exposed to AAV10, which is the vector that we are using, you may have antibodies against it, and that can exclude you. However, if you are one of those that gets treated in a site or get treated during that period of myeloablation, we don't even need to do the antibody testing because I remember I told you that with umbilical cord, your immunity is very, very naive. And so we actually can just administer the gene therapy without even testing for AAV10. However, if, let's say, you're transplanted in a local site and then referred later on to a clinical trial site for treatment, at least 21 days after transplant, most of the time you would be outpatient when you can get referred. Otherwise, you have to do in-hospital to in-hospital transfer. You're going to enter the immune suppressed arm. And that means depending on how much immune suppression you have at that point. So let's say if you're very close to transplant, you're going to be much more immune suppressed than if you're, let's say, 2 years after transplant. You may not be immune suppressed at all. So then we will have to start immune suppression. And what do we use for immune suppression? Well, we have that regimen I explained before, but we also have an independent committee that actually looks at it. And those are experts, immunology experts, gene therapy experts, and transplant experts that look at it and say, okay, for this child who is either receiving already immune suppression or not receiving anything, we think that what the principal investigator or the transplant doctor that is like working with you is recommending is good, or maybe we want to modify it a little bit. So once that recommendation is received, then of course you have signed the consent form and you understand how all the risks of this trial, then the principal investigator or the transplant physician who's doing the trial will go through the qualifying inclusion criteria, again, that you can find in this clinical trial site. And if they fit all the inclusion criteria, then they can be in the trial. Again, for this group, the antibody testing for AD10 is needed if more than 6 months have passed. So if that antibody testing is positive, you would be, at least for the moment, disqualified. Maybe later on you could receive it if those titers go down. But at the moment of the qualifying of inclusion criteria, this is something that can disqualify you. Okay, so what did we learn about those 5 patients that we treated? We learned that we were able to increase the galactocerebrosidase levels, and you can measure this in both in the CSF and the blood, and they would go up. We also saw that there were decreases in cytosine, and those went below what you observe in transplantation. So when you transplant a child, the cytosine levels go down, but they don't necessarily normalize. When you use gene therapy, they do go into the normal range in the myeloablated group. So again, the ones that are treated when they are in the transplant unit. In the immunosuppressed group, it's too soon because it takes a long time for cytosine to actually get into the normal range. So they are going down, but they are not normal yet. Another thing we learned is that in this myeloablated group, you have absolutely no immune response. So you don't have immune response to the GALC or to the vector, which was what we thought we would see, but now we confirm that's true. In the immune suppressed group, we saw some antibodies. And so the effect is not, let's say, as big as what you see in the myeloablated group. What about safety? We didn't have safety concerns relating to the vector. And this is measured by what we call adverse events. And I put it here in capital letters because this is something that creates a lot of confusion when people hear an adverse event. What does that mean? And so I'll go a little bit over what that means. But what we've seen preliminarily is what we call functional efficacy signs. What that means is, what is it that we want to do to help these children, right? So we know that they're having difficulty with motor function because of the progressive neuropathy from the peripheral nerves not myelinating normally. So we really want to change how they function in the motor area, give them more ability to walk, maybe to have more strength, to be more independent. So we're going to call this in general improvements in gross motor development. That's kind of one of the main goals of this trial. So I'll go a little bit of what adverse events are. So in the world of clinical trials, we're regulated by agencies like the FDA in the US and the EMA in Europe, and every country has their own regulatory agencies. But here in the US, is the FDA. And they ask the companies to look for any adverse events. So this trial that we're doing, which is we call it phase 1/2, means we're looking for safety and also preliminary efficacy. So in terms of the safety, that's when we look for the adverse events, okay, how safe it is. So it's quite complicated. So when you look for it, you have to report anything that is an adverse event. And that could be like totally unrelated. It could be that the child had a cold because somebody else in the family had a cold. Or it could be that, the kid just broke their arm and it's totally, is not, let's say, expected. So you have to qualify this adverse event. It could be expected, it could be unexpected. It could be any sign or any symptom. And then in this case, you still haven't figured out if there is a relationship between the study therapy or drug and is it causing it or not, right? So we have to report these. We have to monitor the participant, regardless of what that adverse event is. And then this help us enhance patient care and also evaluate this can actually, an adverse events can affect the effectiveness of the treatment. So it can get very complicated. We have to look at how severe it is. For example, a pre-treatment event. So let's say even though we're evaluating the effect of gene therapy, it may be that something happened with the transplant, right? So pre-treatment, pre-gene therapy treatment, there may be an event. Or there could be, the event could happen after initiating the investigational, in this case, the gene therapy. And so you have to record all this, and you have to record how severe it is. And there are these things called common terminology criteria for adverse events that you have to use and qualify them. Then you have to go and understand if it's expected or not. Why is that important? Because if you imagine a child that has Krabbe disease, that's a progressive disease, which means that you are going to be changing over time, and so you're going to have symptoms. And maybe the treatment is not helping, maybe it is helping, you don't know how much. So there are things that you would expect. For example, you may expect that the child may become irritable if they are developing Krabbe symptoms, but then there will be things that you don't expect. You can also expect things to happen due to the gene therapy, because what's known in other trials that use gene therapy. Or you can expect things that are related to the transplant that are common to transplant. So the causality, what causes an adverse event, has to be qualified by a medical doctor. And this is because you have to have very good medical and scientific judgment and knowledge of not only the disease, but the transplant and the gene therapy. And then there is besides the doctor that is doing the investigation, there is a medical monitor that also provides what they think is what we call the causality assessment. And besides that, there is also the data safety monitoring board. This is a board of doctors that are not part of the company, not part of the trial. They're completely independent. They look at everything. They're usually experts in these areas. And they also say, yes, we agree or we disagree. And all these things take time. And so the reason why I'm explaining all this is that so you understand what it means when you do a trial, what the safety evaluation means. And the effectiveness has to do more with how we can understand if they're doing better than, let's say, what's the standard of care. In this case, would be transplantation. So we do that by doing what's called standardized testing. So if you've been to the NDRD clinic or other clinics that monitor patients, they will be doing some motor testing. They will be, physical therapists are asking the child to like jump, stand on one leg, walk, sit up, all these different things. And you see them that they're scoring these tests. And then you can actually, they do this in the normal population, which would be the gray shaded area. The line in the middle is kind of the average child. And then what you see in yellow is the range of what you see in patients who are transplanted. They are below normal because of what I explained you. Initially, they look more normal, and with time, they become more abnormal. The green ones are the ones that are not treated with transplant. The ones, so I'm talking about the left figure. And this left figure is the 2 kids that were treated as myeloablated. They are in the normal range. Then you can see on the right side, the ones that were treated afterwards. And one is in the normal range. The other 2 are getting better and almost in the normal range. And the blue shaded area on the right shows you kind of the range in the transplanted. So they're better than the transplanted ones. They're still not reaching normal, 2 of them. The one in blue is actually now in the normal range. So you can see that there is a difference in outcomes. But again, we only have 5 kids. We have to do a lot more. So this is going to complete our low cohort. There's going to be a high cohort, and that means a high dose. So we give more vector genomes per kilogram. And we have to do this because we have to understand, how if you give more, do we have a better effect? Or do we get do we get more adverse events? And all this has to be looked very carefully. I'm just going to show you some videos of the kids that were treated. So the one here was 18 months post FBX-101 at 22 months of age. And then there's another one here, 24 months post FBX-101, 27 months of age. So you can see that the one on the right is almost running. So this is a much better outcome than what you typically see from transplant. And then this is a child that was treated in the immunosuppressed group. So this child is having a lot of difficulty here because of weakness in the muscles because of the peripheral nerve disease, standing up. But after treatment, you can see that he's standing much better, and now he gets a little bit silly because he's doing the skill repeatedly, and I think he's actually having fun with it. And now you can see him here running. So these are kind of examples of what FBX-101 has done in some of these patients. So just kind of to summarize, the myeloablative group, which we have treated 2, increases the GALC enzyme activity, both in the CSF and in the plasma, or let's say blood, and it has done it for the period of time that these patients have been followed. There are decreases in cytosine to the normal range and below levels seen when you do just transplant. Again, there is absence of this immune response, and that can cause antibodies against the vector. And they have normal motor function. In the immune suppression group, where there are 3 patients, we also see increases in GALC in both plasma and the CSF. We are seeing the presence of antibodies, and but only against the capsid of the vector, not against the GALC, which is what, again, this because they have the transplant, they wouldn't have antibodies because this immune system has the normal enzyme, so it doesn't see it as a problem. This has translated into improvements in motor function, and the DSMB, so this independent monitoring board, has approved a dose escalation to the, so we're going to be treating now with a high dose. Here I have the same thing, enrollment starting in 2024. We are about to treat our first patient, and this is going to be done in either University of Michigan or Duke University. I would like to acknowledge all our collaborators. There's a lot of collaborators that have been involved even before Forge licenses trial, but these are the ones that have been the most involved. Dr. Mike Gelb, because he's measuring our GALC and cycosine, George Cini, of course, at the Department of Health, Newborn Screening Laboratories, Dr. Mattern, Dr. Poe, who's the senior statistician, the NDRD program that, by the way, now is called Center for Neurogenomics. After I left, they changed the name. Dr. Deepa Rajan is now overseeing that program at the University of Pittsburgh Medical Center. Dr. Sabolch developed the reduced toxicity regimen. Dr. Kurzberg and her pediatric bone marrow transplant team at Duke University were just activated as a site. Also, the University of Michigan, Dr. Vanderlook, the policymakers, the families, the foundations, especially the Rosenau Research Foundation, Krabbe Connect, Hunter's Hope, and the Early Life Foundation. I wanted to tell you that if you want any further information and you are not ready, let's say, to talk to the PIs, but want more information from Forge, please feel free to call Kelly Bozola. She's our patient advocacy member. You have 2 emails here you can use, and Kelly's phone number, which is the one on the right side there in white. I would also like to acknowledge the site in California that was open for a while and followed some of the patients. But right now we just have these two that are active, University of Michigan Medical Center and the University of Duke. That's all I have. I don't know if you have any questions for me now.
Hi, everyone. So put your questions in the chat. Dr. Escolar, I did send you some questions that came through. Do you see those in the chat box?
Let me see. Not yet. No, I'm not seeing them.
Okay. I did send them.
Let me refresh and see if it's no.
Okay. Have your email up too?
Well, let me stop sharing and then I can try to, okay, I stop sharing. No, let me get my email up. Okay. Okay, I have my email up.
Okay. I just sent you the questions by email too.
I haven't received them yet. Let's wait a minute.
One of the questions, Maria, just as a
Okay.
general question.
Yeah, I see them now.
Okay. Okay.
Okay, well, how long does it take to manufacture one dose of gene therapy? So it depends on what vector you're using. There's a lot of things that affect this, but in the case of AV10, it takes a long time, and the reason why, and this could be anywhere from 5 to 9 months, but the reason why is it's because it has to pass a lot of regulatory audits. And so not only it has to, you have to grow these cells and transfect them with what we call the plasmid and then start producing the vector. But just like when you produce any medication, it has to pass a lot of checks. And then it can be administered. It's very, very expensive also to manufacture it at this point. So yeah, so I guess it depends what quantity, how much you need, where it's been made, the experience. We are very fortunate that Forge is a manufacturing company, very, very experienced they're focusing on AAV, and obviously Forge is our well, FBX-101 at Forge is one of our main efforts. So we are very lucky in that sense. There are other questions about how many will be treated with a high dose and what happens when you're done with dosing patients with high dose. So, we have to be continuously interacting with the FDA. Our, let's say, short-term plan is to treat 4, but we may need to treat more. We don't know that. We would find more as we have conversations and our program is evaluated. When would we be done? It depends on the recruitment. We can't predict that because as much as we would like all the patients that we screen to be eligible, as I explained, you have to go through a lot of check marks to make sure that you qualify for the trial. And so it depends, sometimes we cannot predict what's going to happen, how sometimes babies get sick because of the transplant, and that may delay. There is a lot of variables, so it's really hard for us to know the answer to that. But we are trying to go as quickly as we can. We have other question about why is the transplant not part of the trial? Well, because the transplant, so a trial is an investigation of a new drug or treatment, right? So transplant is not new. Transplant is a standard of care. So it's not part of it, because when you have a standard of care that is actually has an effect, a good effect, you cannot, let's say, it's ethically very challenging to tell a family that has a baby with a rapid progressive disease where there is a treatment that can improve the disease, and tell them, oh, would you like to try a new experimental treatment that we don't understand yet if it's going to be effective or not, right? So there is one ethical consideration in terms of not making transplant be part of this. But the main reason why it is not is because it is already standard of care. So we can't make it experimental when it's not, right? What is experimental is the gene therapy. We could have done the gene therapy alone, but we didn't do it because we knew that you need the transplant to have an effect in terms of the cells, the white cells that are needed to produce normal myelin. And so because families are getting the standard of care, what made sense was to infuse them afterwards. It also made sense because of what I explained about the antibodies. Like if you have a healthy immune system that is provided by the transplant, you're not going to reject the gene therapy. So there were many reasons, but the gene therapy was really added after the standard of care.
I sent you another email, Maria.
Am I saying another email? Yes, let me open it. Why is only one patient able to be enrolled at a time? Yeah, that's a good question. So as you saw, we had to do low dose first, right? So usually the process is that because we don't know if we try to estimate how much gene therapy they need based on the animal models, but animals are smaller, they're different, their brains, their peripheral nerves are different size, all that. So there are all these studies that you do to try to estimate what is the right vector genome copies. And so we use that, but we have to be conservative. We don't want to hurt the children. So we try to use what we call the low dose, which is the minimum effective dose that we estimate, would let's say have an effect in the patient. But we have to make sure we're not wrong. So we treat one patient first, and then we review the data. We have to wait many days. We have to collect the data, review it, and then send it to this data monitoring board that then looks at it. And they are the objective group, and they tell us, okay, we've looked at it, we've reviewed it, now you can treat the second patient. So usually after they have given that go ahead, we can treat the second and the third. However, because it's a new therapy, we want to make sure that we don't overwhelm the investigator, because there's a lot, those subjects that have gone through this, they know there's a lot of testing and a lot of coordination. And remember, this is done as an emergency basis in case of the myeloablation. And as you may realize, it's quite overwhelming for a family to hear about the diagnosis of Krabbe and then hear transplant, and then the third thing, gene therapy. The screening procedure requires a lot of different tests that can take 1 to 2 weeks. And then we have to evaluate with blood tests and all kinds of different tests and make sure that we are not causing more harm than good. And then that's the safety part of it. So that's why you see that we are doing kind of one at a time. But as we learn more, and as we've done now 5, we kind of have a much better understanding of the safety. We believe this is a pretty safe vector. And so you may see in the future that maybe there's more than one patient treated at the same time. So we allow that as we feel more and more comfortable with what we're using, because we're getting more and more data that helps us understand the safety of this vector. Is there a shelf life? Yeah, there is a shelf life. We have to keep on monitoring that. So it varies. We are learning about that as we are doing the project. So we keep samples of what was done, what was manufactured initially, and then they get tested, and we make sure that they are still, they're still good. That's part of what the FDA also requires us to do, to make sure that obviously we're using vectors that are still active. Okay, let's see. I think I have more questions. Will we have to close the trial during a period of time? We may have to, or we may not, and it all depends. It depends how
And can you
quickly.
read the full question just so it's
Oh, yeah, sorry.
clear,
Will
please?
you have to close the clinical trial for a period of time when you are done with phase 1/2? How long will it take you to reopen for the next phase 3/4? So we would like to know the answer to that. We don't know it. And the reason why is because, as I was explaining you, how fast we recruit is dependent on a lot of different variables. So, you know, it could take us very few months, or it could take us many months. And so depending on that, we're trying to manufacture as quickly as we can, so that we have enough vector so that we can treat families. But there, as I was saying, the variables are many, even the weight of the children, you know, how big they are, because obviously you require more vector if you are bigger than if you're smaller. And so there's all these things that we have to take into account to understand how much manufacturing we have to do. And because it's a very expensive product, you cannot just manufacture tons of it, you know, and have availability for many, many children. So you have to kind of coordinate both the manufacturing with the enrollment and treatment. So I wish I had an answer for this, but I don't have. There also sometimes are lapses because we need that DSMB board to review data. So for example, after we treat the first high dose, there's going to be probably a month or two where we are reviewing data, we're getting the DSMB approval, and then we will start treating again. So there are always things happening in the background that is making the trial slow down or accelerate. And believe me, as someone who has worked on this for 24 years now, on this treatment for Krabbe, I am more than anyone wanting to treat as many children as I can and go as fast as we can. But again, we have to go. We have to be careful. We have to make sure that the treatment is safe, and that we are enrolling the children that are the best subjects to be able to show that this treatment is either effective or not. We want to make sure that we can in the future benefit as many children as we can, but to be successful, you have to be very careful and design very well the trial and make sure that you know, we don't make mistakes because we're trying to rush too much. Will gene therapy be available eventually for teens and adults, or will the titer be an issue? Our hope is that in the future we can treat all the populations. It's going to take time because these trials take a long time. You know, we realize that this, for example, the mice studies, which is done in the model that recapitulates more the infantile form. You know, we realized this like 2014, 2015, and we are like 9 years later just doing the human trial. So I want to be optimistic, and I want to think that we're going to be able to do this much faster in other populations, but we had to go one step at a time. So we're doing infantile, then we're going to go to late infantile, and then hopefully in the future we can do the other ones, but we are not yet focusing on those presentations. We also think with time, you know, the manufacturing process is going to probably be easier and, you know, maybe less expensive, and maybe we are able to do more for the older populations too. But right now, again, our focus is in the infantile and late infantile.
Jessica, I'll connect you on that other question that's more personal, so offline.
Yeah,
That
so
came
I
through.
cannot answer specific subject questions because of confidentiality. But please feel free to contact our patient advocacy if you have more specific questions about your family members or patients that have been treated already with the gene therapy. Let's see. Can you talk about how the follow-up looks after the clinical trial? So what I mentioned about the follow-up is pretty much how I think it's looking like. I think it's looking very good. The product has been very safe in the sense that we haven't seen the adverse events that are typically seen in gene therapy that is administered intravenously. And I don't have time to go over all the different things that have been reported, but in general, most of these adverse events that are related to the intravenous gene therapy are increases in what we call liver function tests. So the liver gets sometimes a little bit inflamed and some enzymes go up. We haven't seen that. So we haven't seen what other trials have seen. And the reason why we haven't seen it, I think, is because again, it's a very different approach when you treat with gene therapy intravenously a patient who has an immune system and can kind of recognize that and sometimes develop an immune reaction than when you have a normal immune system, which is given by the transplant. You have the tendency to have less reaction. So I think that's one of the reasons why we haven't seen those adverse events. So the trial seems to be quite safe. As with any trial, there are adverse events that can be related to the fact that the children were going to have that. They could be related to the fact of to the disease itself, as I explained, or they could be related to the gene therapy. And evaluating this sometimes takes time. So while I can tell you that it's a safe treatment, we have had adverse events that we have to investigate and are under investigation. So we usually don't release any of the information until we know for sure if it's related to the treatment, because we believe it. Well, we have to report it to the FDA, and we may disclose this. And it is always disclosed to families that are obviously considering treating their children with gene therapy. So part of the consent process is to make them aware of everything that we have seen and their potential relatedness or not to gene therapy. So as I explained in my presentation, it's a complex process because adverse events is considered anything that happens to the child, really, and any event that, it could be that, yeah, while you're walking to the clinic, you just break your leg, or it could be something that we have seen actually in gene therapy, or something we have seen as part of Krabbe or as part of transplant, or something that is just unusual and we don't understand. And so when that happens, we have to investigate much deeply and until we understand it. So the
with that,
Mhm.
Dr. Escobar, just on the follow-up, I think was related to how, if they go through gene therapy, how often do they have to follow up and be seen in the clinic?
Oh,
I don't
yeah,
know if
yeah.
you
Well,
have.
so that's a very good question. And the follow-up is designed in a way that you can capture these adverse events. We want to know if anything is happening to the child. Why? Because we want to know if this could be caused or not by the gene therapy. That's very important for us to understand, because there are more children that will be treated later. So let's say if we find out that an adverse event is due to the gene therapy, it could happen immediately after you infuse them, or it could happen many years later, right? So one, we want to make sure that we pick up the problem early enough that we can treat it. But we also want to make sure that if it is related to the gene therapy, we can consent the next families and explain them what their kids would be at risk for. And if it's something that we simply think is too risky or is too severe, then maybe the trial just stops, or the FDA makes us stop it, or this private data monitoring board thinks, well, that's more risky. Now, in evaluating this, there are many things that interact. Obviously, Krabbe disease is a terribly aggressive disease, but as we know with transplant, we can make it better. So all these things get into consideration. So how long do we follow them up? Well, in our trial, we follow them up for 5 years. However, we have to continue to collect any adverse events reported by the family or doctors for like 15 years. But the families don't have to come in and be participating in the trial more than 3 years after the 2 initial years. So 5 years is total. And it's super important that families get committed to this because we are using an experimental treatment. We don't know what it does long term. And we want to make sure that if there is anything happening, that we can not only treat their child, but also be able to make the other families aware that that could be a risk as they come into the trial. So this is a big responsibility from the sponsor, but also from, I think, the families that consent into the trial. So I would encourage families to continue. The other reason why it's important to continue is that for us to evaluate if this is effective or not long term, we need to see it. If we can't follow the families, we won't be able to see it, and then it affects the entire community, because maybe if we don't have enough follow-up, and that causes the trial not to be successful, right? So there are multiple reasons. I just kind of mentioned some, but these are all the reasons why I would think it's important that families continue to follow up.
Yeah,
Now,
and what you're saying in respect, just to make it clear for families that listen to this recording, Dr. Escolar, is that families can drop out of the follow-ups, that they can opt out of them, but that you're not recommending that because it's of what you've just described.
Yeah. So when you consent for a trial, or actually almost any investigational research, is always voluntarily. You can drop out, and your care will continue as usual, your medical care. You don't have to continue participating. But what I mentioned before is some of the reasons why it's important that you consider continuing to participate in the trial.
Great, thank you. I don't think there's any other questions that have come in, and we are almost at time here anyway. So I just want to thank everyone who participated live today. This is being recorded, so it will be used as a resource to inform and educate those who want to learn more about the ReKlaim clinical trial. I also want to throw a special shout out to Kelly and Dr. Escolar for being here today, for helping educate our community on this opportunity to help improve Krabbe disease. Thank you.
Thank you, Stacy.
All right, have a good day, everyone. Take care.
Bye-bye.
Bye-bye.
Thank you. Bye.